targeting proteins to specific places in a prokaryotic cell. If we think about a prokaryotic cell, there are really only a few places a protein can go. It can go into the cytoplasm, it can go into the membrane, or it can be secreted. In each case, information in the DNA needs to be read out. The first step is transforming it into RNA. This is the process of transcription. In the case of an RNA that's going to direct the synthesis of polypeptides, this process occurs in the cytoplasm. That's the process of translation. Again, where the polypeptide will end up will need to be determined actively. In this case, there's really only a few places to go. It can remain in the cytoplasm, where it could bind to the DNA or act simply in the cytoplasm, or it could go to the membrane. In the membrane, it could be inserted into the membrane or secreted out of the cell. The process by which a membrane protein is made involves what's called the signal sequence. As the newly synthesized polypeptide comes off the ribosome, it interacts with a protein called signal recognition particle. This particle recognizes a signal sequence present in the polypeptide that says, "Take me to the membrane." Once SRP binds the signal sequence, translation halts. The ribosome complex will move to the membrane by diffusion, where it will interact with the SRP receptor. The SRP receptor also acts as a channel, allowing the polypeptide to pass through the membrane. When the SRP is released, translation resumes. The signal sequence is then removed. The polypeptide folds into the membrane or is released into the periplasmic space. Given what you already know, predict what might happen to the final location of a polypeptide if a mutation changed the sequence of the signal sequence.